Welcome, everyone. This is chapter 4, and we're going to be discussing the cell, the difference between a prokaryotic and a eukaryotic cell, the main aspects of the cell, the different parts of it, and then we're going to talk about one of the main functions of the cell, which is, of course, protein synthesis. So let's get started. Okay. Um, so when we're talking about matter, we address the fact that atoms were the fundamental unit of matter in biology and life. The fundamental unit of life, of course, is a cell. Okay. Um, the exception that the author likes to put in here is viruses, but to be honest, viruses cannot be part of the fundamental unit of life because viruses, by definition, are not alive. They don't have metabolism. So cells do have a metabolism. They are alive, and consequently, they are the fundamental unit of life. Let's see. We've This is again a concept that we're addressing again from last chapter. Cells come into existence only through activity of other cells. If you remember, this is a definition of life. Life begets life. Okay. So this is an important concept, and our author likes to reiterate it. Okay. So one of the first things that we're going to delve into here is distinguishing between what's called a prokaryotic and a eukaryotic cell. Okay. So a long time ago, when we have very poor microscopes and we were looking at cells for the very first time in the days of Leeuwenhoek, if you have ever heard of him, he was the first one that brought microscopes to the Royal Society in England. He was a tailor by vocation, but he had an interest in seeing things through a microscope. So back then, the microscopes were very poor. Essentially, they were a single lens. So when they looked at cells for the first time and they found this little round thing in the middle of them, they didn't have the vocabulary to define or explain it, or right. So they called it. It looked like a thimble. I mean, remember who's doing this? It's Leeuwenhoek. What does he do for as a vocation? He's a tailor. He deals with garments. So when he sees this little round thing, he goes, "That looks like a thimble because that's what I deal with every day." Whatever. Anyways, so in Latin, thimble is carry. Okay, carry. So consequently, immediately we started to distinguish between prokaryotic cells. Pro means before. So these are cells that do not have a thimble. What is the thimble that we're talking about? We're talking about the nucleus. So bacteria and archaea, when seen under a microscope, do not have a nucleus. That is not to say that they do not have DNA. They do have DNA. It's just not membrane confined like it is in eukaryotes, and consequently well seen under our microscope to provide what's called a U, or true thimble, or a true nucleus. So those cells that have a well-defined nucleus are called eukaryotic. That's you and me, plants, animals, okay? And prokaryotic cells are those cells that came along before the nucleus, early in evolution, okay? And those prokaryotic cells fall into one of two criteria. They're either bacteria or they're archaea, okay? You and I run into bacteria all day, every day. We drink bacteria. We do. We disinfect bacteria. We deal with bacteria all day, every day. Archaea is a little bit more is a little bit stranger. These are known to be extremophiles. So these are prokaryotic organisms that like to live in very cold places, very hot places, high pH, low pH. Um, what's another good example? High concentration of metals, places where you and I would not like to be. Okay, and you might ask yourself, why would God create such a thing? Sorry, I didn't mean to say it that way. Why would nature? There we go. Why would Mother Nature create such a thing? Well, early on in evolution, um, the Earth was not very nice. The environment in the Earth was either very hot or very cold, or it was very acidic, or it was very basic, or it was very high in metals. So consequently, early in evolution, we had to have an organism that could adapt to those harsh environments, and hence we end up with these extremophiles, which are part of the prokaryotic family. Very, very neat. Okay. Let's see. I don't want to go into this because I'd rather go into um, the things that distinguish prokaryotic from eukaryotic. So I am of the belief that a picture's worth 1,000 words. So these are the main things that and this is not a comprehensive list, but in an introductory biology class, these are the four main concepts that we bring up. Okay. So on the right we have eukaryotic cells. Let's see if I can move. There's always something in the way. Do you notice that? No matter what, there's always something in the way. So on the right, we have eukaryotic cells. On the left, we have prokaryotic cells. Okay. So we usually bring about four main things that distinguishes these cells. Number one, DNA. As we just discussed, in eukaryotic cells, it is membrane bound in the nucleus. In prokaryotic cells, is not membrane bound. As a fact, bacteria have what's called a nucleoid region, where DNA hangs out, but it does not have a nucleus. Okay, it has a little mucoid area where the DNA has a tendency to congregate, but no membrane-bound nucleus. So eukaryotic nucleus, prokaryotic no nucleus. Size, okay? Eukaryotic cells, by definition, are very complex, so they're very, very large. Bacteria and archaea are incredibly small. They are they're hard to see even under a microscope. When we deal with bacteria in microbiology, we have to look at them at 10,000 or 2,000 times magnification, and even then, it's hard to see them very well. Okay, so these are very, very small because they're so simple. Okay, so size: prokaryotic small and simple, eukaryotic big and complex. Okay, organization: eukaryotic cells can be single cell protozoa. They can. However, we know them to form tissues, and we know them to form uh, um, tissue systems, and we know them to make organs, and we know them to build organisms such as ourselves. So every cell in our body forms a tissue. We discussed this, I believe, in the last chapter when we talked about the hierarchy, right? So we went all the way from the atom all the way to the biosphere. So cells come to get to form tissues, and then those tissues come together to form organs, and then those organs come together to form organisms like you and me. So the organization of eukaryotic cells can be quite complex. Okay. By contrast, the organization of these cells, they say single cell. That's such a lie. Okay. These things are actually, they can get pretty complicated. They can even talk to each other, but typically they don't get much more complex than what you and I would call a colony. So make a colony. So it's a whole bunch of cells that can choose to live together. They can choose to live apart. They really don't care, you know. So the organization is very simple here, while in eukaryotic cells, the organization can be quite complex. Okay. And then finally, organelles. You know, we're going to go into organelles in eukaryotic cells. Eukaryotic cells have a lot of different organelles. From a cell biologist, bacteria do not have organelles. Although, if you talk to a microbiologist, they will argue with you. They'll say that they have flagella and cilia and all these other things that they define as organelles. But from a cell biologist, an organelle is a very complex organ. So, in other words, prokaryotic cells, by definition, do not have organelles. Okay, and eukaryotic cells do. Okay, so there we go. To recap, eukaryotic DNA in a nucleus, no nucleus. Size very large, very small, complex, simple. This can go into tissue. This can go into organs, organ systems, organisms. This one usually forms only colonies. Okay, when they talk to each other, they carry out this thing called quorum sensing. You don't need to know about it. And then organelles, eukaryotic cells have many complex organelles. And here the author says one type of organelle. God only knows which one he's referring to. Maybe the nucleoid region. I don't know. Okay. Let's go to the eukaryotic cell because this is what we care about. There are 5 principal components of the eukaryotic cell: the nucleus, other organelles, the cytosol, the cytoskeleton, and the plasma membrane. Okay. So organelles are tiny organs within the cell that carry out specialized functions, such as energy transfer and material recycling. So here we are. What's an organelle? Well, here, let's make this big. Can I move it? There we go. So there's a nucleolus, that's an organelle. There's a nucleus, that's an organelle. There's the rough endoplasmic reticulum, okay? That's an organelle. Here's a lysosome, that's an organelle. Here's a mitochondria, that's an organelle. Here's a smooth endoplasmic reticulum, that's an organelle. Here's a Golgi complex, that's an organelle, okay? And each one of these organelles carry out a different function. So the nucleolus, for example, makes ribosomal RNA. The nucleus confines the genetic material of the cell, okay, via a membrane. The rough endoplasmic reticulum is responsible for accepting ribosomes and producing protein in here, an event called translation that we'll talk about in just a little bit. Mitochondria are responsible for the production of energy. Lysosomes, very important. This is the cleaners of the house. Okay, if there's something around that needs to be destroyed because we need to get rid of it, these lysosomes have the enzymes necessary to break everything apart and get rid of it. So this is our waste management here. The Golgi complex, oh, very important. This guy's responsible for shipping. Okay, we'll go into that in just a little bit. Here's our smooth endoplasmic reticulum. This is responsible for making essential fats in the cell, and it's also essential for the elimination of toxins in the cell. So, for example, hepatocytes, the cells in the liver, are rich in smooth endoplasmic reticulum because they deal in the elimination of many wastes. Very, very neat. Okay, so all these are organelles. Feel free to address those on your own. We're going to go into it in a little bit more detail in a second here, but can't hurt for you to look at them either. Let's see, the cytosol is a jelly-like fluid outside the nucleus in which these organelles are immersed. This is important, okay? Students love to confuse the word cytosol with the word cytoplasm. These are two different animals, okay? Just as this is, it is outside the nucleus in which these organelles are immersed. In other words, the cytosol outside the nucleus, inside the plasma membrane, and then exclude all the organelles, and that is the cytosol, okay? By contrast, The cytosol should not be confused with the cytoplasm, which is a region of the cell inside the plasma membrane but outside the nucleus. So in the cytoplasm, you go outside the nucleus, you go inside the plasma membrane, you include everything, include the organelles, and that is the cytoplasm. You exclude the organelles, and that is a cytosol. Okay, that's important to know. The cytoskeleton is a network of protein filaments. It functions in cell structure, cell movement, and the transport of materials within the cell. So, and we're going to go into detail on that in just a second. The plasma membrane is the outer lining of the cell. We discussed the monomer of the plasma membrane last week. Remember, we said that it was a glycerol with 2 fatty acids, a phosphate group, and a variable group, which consisted of a sugar, okay, for cell recognition. So here you have it, the different parts of it. The nucleus, bam. The organelles, we went over some of the organelles. The cytosol, notice that we've excluded the nucleus, everything inside the plasma membrane, and we have excluded the organelles, that is the cytosol. The cytoskeleton, there's 3 aspects to the cytoskeleton. We're going to go into that in detail in just a second. And then, of course, the plasma membrane. So these are the main components of the cell. Okay, now, I don't know about you guys, if you've had biology before. I had biology when I was in elementary school. And what our teacher did, which I thought was very neat, is she made a metaphor between the cell and a factory. She said at the end of the day, every cell is like a little factory. So he's making something. Not a bad statement. As a fact, that describes what we're going to hit here in 4.4 very well. Okay. This is going to be a tour of the animal cell's protein production path. Information for the construction of proteins is contained in the DNA located in the cell nucleus. So this can be a factory. Another analogy that I use is you have a friend, and he or she asks you to make a I don't know, a pineapple, upside down pineapple cake. I've never had one. I don't even know what one is, but that's what came off my mind. An upside down pineapple cake. And he or she has it, and they just think it is marvelous. It is marvelous. So he or she asks you for the recipe, okay? Now, you guys are pretty young. Way back when I used to cook or bake, we used to have these things called Rolodexes. You guys probably haven't even heard of these things. It was this round thing, and you went through it, and it had these little 3x5 cards, and those 3x5 cards had your little recipe on there, right? Now, you guys don't do that, right? Because you guys don't even know what a Rolodex is. You guys probably, what you do is you have all your recipes on your computer, or you have it on your cell phone or whatever, right? But here's the thing. You like your friend, you trust him or her, but would you give them the original recipe and go, here you go, have fun, good luck, give it back to me when you're done? Well, I don't know about you, but I wouldn't do that, even if I trusted the person, okay? So what do you and I do? What would I do? What I would do, and this is crazy, I would go to my little Rolodex where I had my recipe, and then I would grab a piece of paper and pen, and I would copy the recipe down, and then I would give that note to my friend, and they wouldn't have to touch my Rolodex, which has my very valuable recipes in it. Turns out that the cell works the same way. We're going to call its little Rolodex or its cell phone, we're going to call that the nucleus. Okay. So what we have, let's go back here to the picture. Here's the nucleus, right? And I don't like this. I want a nice, beautiful picture of my, that's perhaps as good as it's going to get. Okay, so there's my nucleus. There's my Rolodex. There's my cell phone with all my information, which I'm not going to give out because it's very valuable. I don't want anybody to mess it up, right? So instead, what did I do? I jotted it down on a piece of paper and I handed it out. The cell does the same thing. It takes its DNA, which is inside this computer, and the DNA never leaves because it's too valuable. It's the original copy, if you will. So what it does is it copies the DNA into a piece of paper, which is called, surprisingly enough, messenger RNA. And then that messenger RNA can go into the cell. And what it is an instruction set that tells the cell how to put together things together. Okay? I want a bigger picture of my cell because I don't even want to follow my PowerPoints right now. I just want a nice little cell. What does it take to get a nice little cell? Here, okay. So once it makes a messenger RNA, the messenger RNA slips out of a pore, out of the nuclear envelope, and then attaches to a worker. I mean, somebody, it's a message, right? It's got instructions. Somebody's got to do it. That somebody in the cell is called a ribosome. The ribosome is a little guy. It's an enzyme. It's a protein. And is the one that can read the messenger RNA, and it gives it instructions to assemble amino acids in a very specific way. We've kind of alluded to this already, right? So once that ribosome gets that piece of messenger RNA, depending on where that protein is going, the ribosome will remain in the cytosol, not in the cytoplasm, in the cytosol, or it'll attach to this beautiful little machine called the rough endoplasmic reticulum. Now, why is it rough? It's rough because it's littered with ribosomes all doing the same thing, producing a protein. Okay? Endoplasmic is inside the plasma membrane. Okay? Reticulum means net, is Latin for net. So this is a net that's inside the plasma membrane that is rough with ribosomes. Ribosomes being the cute little workers whose sole purpose is to read that little note we jotted earlier, which is called messenger RNA, and attach itself to this RER, this rapid endoplasmic reticulum, and produce a lovely little protein. Is that cool? Very cool. Now let's see how our author tells us. I should follow my author because that's what keeps me from making mistakes. Okay, so this information is copied onto a length of messenger RNA, mRNA, that departs the cell nucleus through its nuclear pore, and then goes into the sites of protein synthesis called what? That's right, they're called ribosomes. And these lie inside the cytosol. I don't like cytoplasm. They're in the cytosol. It's not on an organelle. It's off an organelle, okay? Many ribosomes that receive the messenger RNA change process only a short stretch of them before migrating to and then embedding onto a series of sac membrane networks called the rough endoplasmic reticulum. Okay, the rough endoplasmic reticulum is responsible for proper protein folding. I got to tell you, this is not easy, not easy at all. Nobody tells you this. It took me going to graduate school to find out that under the best of conditions, only about 30% of the protein is folded correctly. Can you imagine that? If you and I had a factory producing pineapple upside down cakes and only 30% of them came out right, they would fire us on the spot. But this is a good day for the cell. What does this convey to you? What does this tell you? What this tells you is how hard it is to fold a protein correctly. It is not easy. This is hard. Okay. Now, as if that weren't enough, let me move my thing here. The quality control of this protein folding is also carried out by the rough endoplasmic reticulum. So if it's folded properly, it keeps on going. If it's not folded properly, it comes back to be degraded, okay, to be broken apart. By who? A lysosome. We talked about them earlier. So this is very cool because as we're making these upside down pineapple cakes, it's got a factory feel to it. And you can picture a factory, you know. You got the guys with the blueprints inside the cell nucleus, and then they jot everything out onto messenger RNA, and then it goes down a conveyor belt. That conveyor belt is a rough endoplasmic reticulum, and you can picture that as it goes through, that protein is getting longer and longer and longer and longer, right? And the reason I like that picture is because that's exactly what's happening. As a matter of fact, it's called the endomembrane system. Endo meaning that it's inside the cell, and as a membrane, it's a system. It's not happening by chance. Everything is being controlled very specifically. Okay. The endomembrane system consists of the rough endoplasmic reticulum and the Golgi apparatus. We're going to hit that in a second. So quality control is taking place at the rough endoplasmic reticulum. If things aren't folding correctly, and the rough endoplasmic reticulum has proteins inside of it, your author doesn't cover them. It's beyond the scope of this class. They're called chaperone proteins, and those chaperone proteins are supposed to facilitate, aid, help in the proper folding of these proteins. Okay. Okay. So, sequences are dropped from ribosomes into the internal spaces of the rough endoplasmic reticulum. The ribosome is still attached, okay, still producing that protein. It just happens to be tethered. It's on the surface of that rough endoplasmic reticulum as the rough endoplasmic reticulum moves down the conveyor belt via this endomembrane system. There, the polypeptide chains fold up, thus becoming proteins and undergo editing. And so we have this, what's called the central dogma. In the central dogma, we say that we have DNA from the nucleus that gets transcribed into messenger RNA in the nucleus that gets translated into protein by the ribosome, okay? And it can either be a free ribosome or it could be an attached ribosome on the RER, okay? So the area inside the nucleus devoted to the production of, the nucleolus. My apologies. We're talking about the nucleolus. Let's see if we can't go to it. See the nucleus here? And there's that little black thing in the middle. That's called the nucleolus, okay? And the nucleolus is responsible for the production of ribosomal RNA. So this is responsible for producing that RNA that's going to be used by the ribosome itself. This is what the ribosome is made out of, ribosomal RNA and protein. Okay, some ribosomes are not embedded into the rough endoplasmic reticulum, but instead remain freestanding in the cytosol. The significance of this is that the protein that's being produced by this uh, um, ribosome is not going to be transported anywhere. It's going to be produced on site to be used on site. If the protein is being produced in the rough endoplasmic reticulum, it means that it needs to be transported to some place specifically. It might be a membrane inside the cell, it might be the plasma membrane, or it might be outside the cell. Okay, so for example, if you're making insulin, that insulin is a protein, and that insulin, once it gets to the Golgi complex, is going to be sent to the outside of the cell so it can be used by the body. Okay, but if the ribosome is not attached to the rough endoplasmic reticulum, if it's a free ribosome, then again, what that means is the protein that's being produced by that ribosome is going to be produced right there and is going to be used right there. Okay, so materials move from one structure to another in the cell via the endomembrane system. Okay, here a piece of membrane with proteins or other materials inside can bud off from one organelle, move through a cell, and then fuse with another membrane-lined structure. Why do we care? Because so far we have only talked about the rough endoplasmic reticulum, okay? But all the RER does is it produces protein. Once you have that protein, once you have your pineapple upside down cakes done, okay, you have to ship them off. Otherwise, why make them, right? So what that means is that they get put in little boxes. They get put in little boxes, okay? Those little boxes are called transport vesicles, okay? And they go from the rough endoplasmic reticulum, and they enter what's called the Golgi complex. What's the Golgi complex? It's FedEx, it's UPS, it's the U.S. Postal Service. This is a guy that um, modifies the little box that we made, this transport vesicle, by adding sugars, by adding phosphates, by cleaving proteins. In other words, it's putting an address label on there. This is where you're going, okay? And then it gets transported via the Golgi complex, okay? They are further processed and further marked for shipment to appropriate cellular locations. Isn't that cool? Okay, so here we go. Transport vesicle from the rough endoplasmic reticulum fuses with the Golgi complex. Proteins undergo more processing in the Golgi. Side chains are edited, sugars may be trimmed, phosphate groups may be added, proteins may be cleaved. God only knows. All we're trying to do is we're trying to do those things necessary to finish the product and get it shipped in the right direction. Okay, and then where do they go? Well, they might go to some place inside the cell, or it might go somewhere in the cytosol, or it might go to the plasma membrane, or it might go to the outside of the cell. Okay, now these little folds, we love the word cistern. Biologists love the word cistern. These little folds are called cistern. Cisternae is plural. Okay, this is a cisternal space. You and I have never heard of a cistern. The reason you and I have never heard of a cistern is because we live in modern times. A cistern in the old days, when they needed water and they were afraid that they might not have water, what they would do is they would dig these deep holes into the ground. No, they're not wells, they're cisterns. and then they would cover the inside of these holes of these spaces with special rock and covering that was impermeable to water. That way, when it rained, the cistern would fill up with water and the water would not go anywhere, and these people would have somewhere to drink water from. Okay? That's called a cistern. It is not a well, it's a cistern. Well, in biology, anytime that we have a little crevice like this, we love to call it a cistern. Why? Because it's full of something, probably water. It fits, you know. Okay. Other items. The smooth endoplasmic reticulum is a network of membranes that function to synthesize lipids and detoxify potentially harmful substances. Why does it have to synthesize lipids? Here's the thing, guys. Remember how the plasma membrane, again, has got two fatty acids, it's got a glycerol, it has a phosphate group, and then it has that variable group, which is sugars. And do you remember how I told you that that variable group was like a little ID card that let the cell go, hey, I belong here. Don't arrest me. I'm actually here. I'm supposed to be where I'm at. Okay. So what this means is that the smooth endoplasmic reticulum is always producing uh, lipids for membranes, both inside the cell and for the plasma membrane that allows these membranes to identify themselves to each other and to our immune system. Okay, so this smooth endoplasmic reticulum is very important for the synthesis of these lipids. It's also responsible for the detoxification of harmful substances. So, I mean, I used to be a medic, you know, and I'm not happy about this, but one of the things that I had to deal with was suicides. Okay. And a lot of things that people would do is they would engulf medication, not knowing what the medication would do in order to you know, render themselves lifeless. And you know, they would do stuff like Tylenol and aspirin and stuff like that. And we would pull them out of the brink of death by lavaging their stomach. But you know, we would have a conversation afterwards, and that conversation is, you know, you think that you escaped dying, but honestly, what you have done is you have injured your liver and your kidneys by taking a medication to a much higher concentration than you should have. And this is terrible because you know what? Tomorrow you're going to decide that what life is worth living, and your liver is not going to agree with you. So anytime that you consume something that is toxic, it doesn't have to be an excess of aspirin. It could be an excess of alcohol. Okay, the liver is beaten up, and uh, and the kidneys are beaten up, and that's limiting your lifespan. And the reason for that is because the liver's hepatocytes are rich in this smooth endoplasmic reticulum, whose sole purpose is to void the body of toxic substances. Okay. Lysosomes. Oh my gosh, these guys are so important. These are organelles that break down worn out cellular structures or foreign material. So if you have a question on an assessment, just hinting, and it's asking, hey, this thing is supposed to break things down and get rid of them. Chances are the answer is lysosome. Okay. Lysosomes are incredible. What they do is they have an enzyme that breaks down everything. They break down DNA, they break down proteins, they break down lipids, they break down saccharides. So when a lysosome attaches itself to a vacuole that contains something that shouldn't be there, that shouldn't be there is not going to be there for much longer, because that lysosome is going to destroy that item. Okay. So lysosomes are incredible. They're our waste management. Okay. and they break down everything that's not supposed to be inside a cell. Once the digestion is complete, is left as monomers that can be reused by the cell. So quite incredible there too. Okay. Now, I love this. I have long criticized this this slide, and yet here it is. I I find it very entertaining. Um, so here is, according to the author, here's a lysosome digesting a mitochondria because mitochondria is not working properly. I'm going to let you in on a little secret. In real life, this is never going to happen. And the reason for that is because if anything, it doesn't have to be a lysosome, but if anything attacks this angry little mitochondria here, and this mitochondria's membrane ruptures, and one of his proteins called cytochrome C comes out, it is going to produce one of the most powerful destructive machines known to a cellular body called an apoptosome. If you don't believe me, Google it. This is cool. The apoptosome is so powerful that it will liquefy a cell in probably less than 10 minutes. I mean, there'll be nothing left. So lysosomes might digest and destroy a lot of things in the cell, but this is a poor choice of an organelle that it would go after. Okay. If you have a misfunctioning mitochondria, it will probably lyse and destroy the entire cell anyways. So I like this. You know, all this demonstrates is that because something can be drawn, it doesn't mean that it's true. It's cute. Okay, mitochondria and energy. Mitochondria are organelles that function to extract energy from food and to transform this energy into a chemical form the cell can use, the molecule adenosine triphosphate. And this is key to this term because that's going to be the heart, the core of this term. Okay, the core of this term is going to be glycolysis, the Krebs cycle, and the electron transport chain. And so a lot of this is taking place inside this beautiful organelle called the mitochondrion. Okay. Another thing that's very interesting that you should remember is that mitochondria contain some of their own DNA. Okay. And so this relates to a concept called endosymbiotic theory, where we believe that eukaryotic cells captured mitochondria and plant eukaryotic cells captured chloroplasts, and that mitochondria and chloroplasts were once free living organisms that have since become indentured servants of eukaryotic cells. And, you know, for a long time we thought that this was baloney until we actually found mitochondrial DNA. Now, don't get me wrong, the mitochondria cannot live on their own anymore. And the reason for this is because during the symbiosis of evolution, a lot of the mitochondrial DNA has migrated to the nucleus of the eukaryotic cell. So the mitochondria now no longer has all the genes it needs to function independently of the cell. But the fact that it has its own DNA implies that at one time it did. So this concept of endosymbiotic theory is incredible because it reinforces a concept that evolution is uh, a true concept. I mean, if you don't find that mind blowing, I don't know what you will find mind blowing. Okay, so again, to recap, both mitochondria and chloroplasts have some of their own DNA. It has not all migrated to the nucleus, and as a consequence, that implies that there were once free living organisms. And this concept is called the endosymbiotic theory. Very, very neat. Is that beautiful? What a beautiful little guy. It's sausage shaped, It has an inner and an outer membrane. The majority that we're going to be studying this term is right between these two membranes. This is a magical organelle, just magical. Oh, arguably my favorite. Okay, and then we have these little convolutions here. What do you think they're called? Cisterns? No, they're called cristae. Isn't that hilarious? All this time to learn that anytime they have a little space, it should be called a cistern. Yeah, in here they're called cristae. So these little convolutions are called cristae, and the purpose of the convolutions is to maximize surface area inside the mitochondria. Oh, what does the mitochondria do? It produces energy, right? Glycolysis takes place out here, and then we have the Krebs cycle in the matrix, and then we have the electron transport chain right here in the inner mitochondrial membrane. Very, very cool. So it harvests and produces energy, just like the chloroplast. And we're going to stop right there to give you a little break, okay? Or give me a little break, or give somebody a little break. See you back in a bit. Go to the bathroom, grab a drink of water, get some coffee.